For over a century and a half, the true origin of Neanderthals was buried beneath legend and guesswork. Then ancient DNA from a Spanish cave exposed a genetic twist no one predicted. In 2013, scientists found Denisovan ancestry in bones that were thought to belong to Neanderthal ancestors, and the discovery upended everything. So what do these ancient remains really reveal, and how did Europe become the cradle of a lost human lineage? A narrow shaft plunges 13 meters into the limestone heart of the Atapuerca Mountains. At the base lies Cima de los Huesos, the pit of bones, a chamber unlike any other in the study of human origins. Here, more than 7,000 human bones have been recovered, representing at least 28 individuals who lived and died roughly 430,000 years ago. The remains are jumbled together, layered in ancient sediment, their preservation owed to the cave's constant cool temperatures and a stable, mineral-rich environment. This natural vault protected even the most fragile bones from decay, creating an unparalleled archive of early human life. Excavating Cima de los Huesos has never been simple. Researchers descend by rope, navigating darkness and narrow passageways to reach the chamber below. Each field season, teams led by Juan Luis Arzuaga, Jose Maria Bermudez de Castro, and Eudald Carbonell carefully sift through the sediment, mapping every fragment. The scale of the discovery is staggering, not just for the number of bones, but for their completeness. Skulls, jaws, limb bones, and even delicate inner ear structures have been recovered, some belonging to adolescents and young adults. The concentration of so many individuals in one place is a mystery in itself, fueling debates about whether the pit served as a burial site or a natural trap. Among the scattered bones, a single artifact stands out, a finely crafted hand axe shaped from red quartzite. Named Excalibur, it is the only stone tool found in the chamber, unused and almost ceremonial in appearance. Its presence suggests a level of symbolic thought rarely attributed to such ancient humans. The hand axe, alongside the remarkable preservation of the remains, has transformed Cima de los Huesos into a focal point for questions about human ancestry and behavior. With so much physical evidence at hand, scientists saw an opportunity to look for genetic traces that might finally resolve the origins of these early Europeans. In 2013, a small team at the Max Planck Institute for Evolutionary Anthropology set out to attempt the impossible. Led by Matthias Meyer, they worked in ultra-clean labs, armed with new techniques built for the most fragile, degraded DNA on Earth. Their target was a single femur, buried for nearly 430,000 years at the bottom of Cima de los Huesos. No one had ever recovered hominin DNA this old. The odds of finding anything readable were slim, yet after months of painstaking extraction and sequencing, Meyer's team pulled out an almost complete mitochondrial genome, the oldest human DNA ever recovered. The result sent shockwaves through the paleoanthropological world. The bones from Cima looked like early Neanderthals, and the site was deep within Europe. By all expectations, their mitochondrial DNA should have matched Neanderthals, but the data told a different story. The Cima mitochondrial DNA clustered not with Neanderthals, but with Denisovans, a mysterious human group known only from a handful of fossils in a Siberian cave thousands of kilometers away. This unexpected link raised immediate questions. How could individuals in Spain, whose bones showed clear Neanderthal features, carry a maternal lineage more closely related to Denisovans? Was there an ancient connection between these populations? Or had the Neanderthal family tree been more tangled than anyone realized? Some researchers wondered if the Cima people were ancestors to both Neanderthals and Denisovans. Others speculated about an ancient population that had split, leaving its genetic traces scattered across Eurasia. The only certainty was that the discovery had upended the neat evolutionary trees drawn from bones alone. For Meyer, the contradiction was more than a puzzle. It was an open invitation. The mitochondrial genome offered only the maternal story. To truly understand where these ancient Europeans fit, his team would have to push even further, seeking answers hidden in the nuclear DNA. Inside the Max Planck Institute's ancient DNA labs, every surface is sterilized, every tool flame treated, and every breath filtered through masks. The challenge is absolute. Recover genetic material from bones that have lain in the earth for over 400,000 years. Under the guidance of Svante Pääbo, the team developed protocols that would become the gold standard for ultra-ancient DNA. No ordinary laboratory could handle this task. Even a single skin cell from a modern researcher could overwhelm the faint molecular traces left in the fossils. Samples are moved in sealed containers, and technicians work in positive pressure clean rooms, their suits reminiscent of a hospital operating theater. The bones themselves are selected with precision. The dense petrous portion of the temporal bone deep within the skull is prized for its ability to shield DNA from water and microbes. Dental pulp, hidden inside teeth, offers another safe haven. Researchers use drills to reach untouched inner surfaces, extracting fine bone powder that holds the last remnants of endogenous DNA. But what they recover is not a pristine double helix. Instead, the fragments are broken, often less than 50 base pairs long, and riddled with chemical scars. One of those scars is cytosine deamination, a chemical change where cytosine bases turn into uracil, then appear as thymine during sequencing. This pattern, especially at the ends of DNA fragments, becomes a signature of authenticity. Modern contamination lacks these marks. To further guard against error, every extraction is repeated independently, and sequences are compared for consistency. Single-stranded library preparation, a method pioneered by Meyer's team, allows even the shortest and most damaged molecules to be captured and read. Magnetic beads bind to DNA, pulling it from a soup of bacterial and fungal contaminants. The work is slow, the yield is minuscule, but the payoff is enormous, each molecule authenticated, each base call scrutinized for ancient damage. The result is a data set that can be trusted. The methods developed here, targeting petrous bone, exploiting characteristic damage patterns, and using ultra-clean protocols, set the foundation for extracting not just mitochondrial DNA, but the far more elusive nuclear genome. For the first time, the question of Neanderthal origins was no longer a matter of speculation. The technology was ready to deliver answers from the heart of the Sima de los Huesos. In 2016, the Max Planck team achieved what had seemed impossible: nuclear DNA from the ancient bones of Sima de los Huesos. This was not just a fragment, but a genetic blueprint from both maternal and paternal lines, allowing researchers to place these individuals with unprecedented certainty on the Neanderthal branch of the human family tree. The nuclear evidence left no doubt. These were not Denisovans or some ambiguous cousin, but early Neanderthals, the direct ancestors of the classic Neanderthals, who would later dominate Ice Age Europe and Western Asia. The genetic evidence forced a dramatic revision of the evolutionary timeline. Counting the slow accumulation of mutations, scientists calculated that Neanderthals and modern humans last shared a common ancestor between 550,000 and 765,000 years ago, far earlier than most fossil-based estimates. The Sima de los Huesos people, already showing Neanderthal features 430,000 years ago, stood as a testament to this deep split. Their nuclear DNA also revealed a story of survival against the odds. Neanderthal populations were never large. At their peak, only about 3,000 to 10,000 breeding individuals likely roamed the vast glacial landscapes. Such small numbers made them vulnerable to genetic bottlenecks, as climate swings and shifting habitats repeatedly squeezed their populations. Yet, within their genome, researchers found clear signs of adaptation. Genes tied to brain growth, immune defenses, and the ability to withstand cold were already under strong selection in these early Neanderthals. Physical traits, broad noses, heavy brow ridges, and powerful builds were not simply accidents of fate, but genetic responses to the harsh environments they faced. The nuclear DNA from Sima de los Huesos closed the case on their identity and origin, and it also opened new possibilities. Recent advances now allow scientists to extract ancient DNA directly from cave sediments, even without bones. As these methods spread to new sites, the genetic map of our ancestors grows ever more detailed, promising further surprises in the story of human evolution. Every living human outside Africa still carries Neanderthal DNA, proof that their story is also ours. As genetic tools sharpen, the boundary between them and us grows ever thinner. The ancient past isn't distant. It is written in our cells. What will your DNA reveal next? Share your thoughts below.